The last thing we talked about is we were talking about the cytoplasm. It acts like a soup of life. Basically contains all the essential enzymes and proteins that are responsible for many of the metabolisms that take place inside the cell. You're looking at intermediate metabolism such as carbohydrate breakdown or uptake. It also takes place in cytoplasm, ribosomal protein synthesis. These are essentially simple proteins that you're making, which are usually enzymes. Ribosomes that are bound to membranes end up doing complex proteins that we end up using as lipid membrane proteins. So that's a little bit different in the sense that the ribosomes are actually purposely bound and incorporate the protein as it's being produced into the lipid membrane. Usually that takes place in what's called the rough endoplasmic reticulum, which I'll show you here in a few minutes. Okay? But the essential protein factory that's found in the cytoplasm is a free-floating ribosomal protein matrix. It takes cytoplasmic metabolism products and it does store it as inclusion bodies, either as carbohydrates, fats, and sometimes will form secretory vesicles, which means that it empties waste products out, usually into the interstitial space where the body then gets rid of it through the lymphatics. The eukaryote organelles are very important in the way they're structured. This is the analogy of the city. Okay? The first things that we're going to talk about are going to be all these things, which is the nucleus, ribosome, endoplasmic reticulum, Golgi, lysosomes, peroxisomes, mitochondria, vaults, and cytoskeleton. These are all basic structures you need to know because we come back and talk about this in anesthesia as to different effects and how things are metabolized in the cells. The first structure, the most important one in the sense of what makes you who you are, is the nucleus. The nucleus is, if you want to think about a repository of information, is just take the Library of Congress of the United States and all the books that it has in it. That's pretty much the equivalent to what's inside your individual nucleus. Just think of that for a second. Think about how much information that means it's carrying inside there, okay? And how compressed it is, okay? So what you're literally doing is you're taking a giant library, okay, and you're compressing it down to a very, very small encoding mechanism so you can actually build a human being out of it. Okay? And that's what a nucleus really does. It contains all your chromosomes and your DNA in it, so it makes and replicates every single cell in your body. Okay? Now, your body selects what it's going to read. Okay? Now, the important part of the nucleus is that it has to hold all the repository of information intact. It cannot let it free float in the cytoplasm or it can get injured. Okay? It's got to hold it into a very tightly packaged area, which is inside the nucleus and nucleolus. Okay? So what I want you to think of is imagine it's a giant library, okay, where the library is all reference books. There is no book that you can check out. Pretty much a little bit like our library. Okay? You have a bunch of books, but you can't really check them out. But you do have something right next to it. It's called a printer, okay, or a photocopier. The process basically does the same thing. It takes reference material that you want, and then you can take it to a copying machine and copy it. Okay? And it's still, that's considered a transcription. The reason it's transcription is you're not changing the language. All you're doing is you're just taking the book from being a reference book to something that's transportable, which we call a photocopy. In this case, a photocopy is called messenger RNA. Okay? So it goes from DNA to be converted via transcription to messenger RNA. Okay? But they're both, DNA and RNA are still the same language. Okay? They are A, which is deoxyribonucleic acids, and the other ones are ribonucleic acids. So they're both a form of nucleic acid. Okay? So they're both talking English. Okay? So that's why it's called a transcription. You're transcribing it from one book to another. Okay? The DNA stays always inside the nucleus. It doesn't leave. But the messenger, on the other hand, leaves. Okay? That can be taken home. So if you wanted to build a car and you wanted to build a door on the car, you would just have to go to the book for talking about car doors. You photocopy it, and then you take it with you outside so you can do your work. And if you leave it and the oil gets on it and it's damaged, who cares? Yeah, I'll get another copy. But if I take the DNA out and damage it, That's really bad. That's losing your reference book. Okay? So that's what you don't want to do. So things that will cause damage inside of DNA will be ionizing radiation, okay, which we work exposed to a lot in the OR. So eventually, if you get enough radiation exposure, you will cause damage to your DNA, which you don't want to do. Okay? Some other molecules also will penetrate and cause damage, and that's why we tell you stay away from it if you're pregnant in certain conditions. The very important structures here that actually hold the DNA together is a thing called histones and chromatids. Okay, we think that's related to aging. Okay, we actually are beginning to truly believe that. It was when I was first starting out, we used to talk about what causes us to age. And we think it's somehow related to this chromatin strand, how tightly it's bound. One of the things that everybody sort of thought was, if you take an animal and you clone it from one animal and you make the exact copy from that DNA and make it, okay, the animal should live a normal life. And it doesn't. And that's when I became very curious as to why. So if you had a sheep, let's say and the average lifespan for a sheep was, say, 7 years. And you took the sheep at 2 years out, okay, and you made a clone of him. Okay? The clone would only live 5 years, and he would drop dead. So I was like, wait a minute, why is that happening? Well, what's happening is the chromatid strands that basically are like a time sequence, they start unwinding off the DNA. The looser the chromatin strands are, the more DNA damage you have, and the more intrinsic error you have, so the cells begin to die. Okay? So all of us have a way that our chromatids unbind at different rates. If you have progeria, it means you had an advancement of that chromatin strand displacement, the person will age very rapidly. And even though they're only 6 or 12 years of age, they look like they're 90 years old. Okay? Now, just think of that. That means reverse can be possibly true. And we think we know that we have certain people, like say in France, that live in the Alps, that live to 105, 110 years of age. That's average for them. Why? What the new studies are showing is that the chromatin binding may be inhibited by something that they're eating in their environment. And if they can figure out what that is, we'll be sucking down pills of chromatid stabilizing molecules and we'll be living for 200 years, right? Okay, great. All right. It's one more problem to deal with. But it'd be kind of cool. That means you could actually study anesthesia for 10 years and not worry about it. Because you can live for 120 years, so what? So, no, I don't want to do that to you. All right, now, the messenger RNA, as it travels out into the cytoplasm, it can go two things. It can go two places. It can go to a ribosome that's free-floating so that I can work and produce something that I'm going to dump in the cytoplasm, or I can work right against what's called the endoplasmic reticulum, where then I'm going to dump the protein directly into a membrane. Okay? So that's only two places it's going to go. Okay? The RNA complex is basically translated now. Why is it translated? Because it's converting it from English to French. Okay? It's no longer nucleic acid that's forming. It's now switching the conversation to amino acids, which eventually form proteins. So I'm going from nucleic acids to amino acids. So that's a translation. The process of doing that, identifying it, is by using 3 of the messenger RNA molecules, okay, which is AT, I think AT and CG, okay, and then in this case it's AU and CG. So that's inside AT-CT is the CG is the DNA. So this is RNA. When you translate this information, it's basically identifying a 3-subset codon to identify which amino acid I'm going to put on there. Okay, so I literally will stack an amino acid for every 3 encoded codon here by the messenger RNA. Okay? And eventually builds this a protein. I don't want you to know all the details of that. That's not necessary for your job. What you need to understand is that messenger RNA is going to be translated into amino acids. Okay? Eventually, this will lead to enzymes that are freely floating inside the cytoplasm. If, on the other hand, I want to incorporate this into, let's say, a cell membrane, I have to attach the ribosome to the endoplasmic reticulum, which ends up being called the rough endoplasmic reticulum. And that actually produces a complex protein synthesis factory of the body, of the cell. So the endoplasmic reticulum is really the complex manufacturing system. That's really your factory of all the big proteins that you're wanting to make. Okay? There's two kinds of endoplasmic reticulum. There's smooth and there's rough. The smooth is for the production of lipid bilayers. Okay? These are smooth. Okay? And then when you start embedding proteins in it, it looks like what? It becomes rough. When you look at an electron microscopy, it goes from a smooth lipid bilayer to a rough-looking thing with a bunch of proteins embedded in it. Okay? So the smooth endoplasmic reticulum is where you're putting your lipid bilayer together, and the rough is where you're producing your proteins that are embedding into the lipid bilayer. Now, once you make all this stuff, then it goes from there, and it gets processed by another company. Okay? So it's literally, if you make your fancy car with all the doors on it, but you forgot to paint it. Okay? The actual painting department and the cleaning up department before it gets deposited is the next system, which is a Golgi apparatus. Okay? The Golgi apparatus is your refining and packaging company. Okay? It's taking all the final byproduct of the manufacturer and it's really making it so it can actually sell it. Okay? Which means it takes information like lipid bilayers and sort of arranges them improperly and then forms secretory vesicles which then can bind and cause the protein to be where it should be, inside the cell membrane. Proteins from the endoplasmic reticulum are packaged in the Golgi complex. And the actual membrane, the actual protein that you want facing outside, when the Golgi releases it into your cytoplasm, is actually inside. Okay? So it's a completely sealed, here's how I want you to think of it. Okay? You're a secretory vesicle coming out, and my face is what I want facing outwards. Okay? So I'm inside the vesicle right now. As soon as I bind to the lipid bilayer, I separate this vesicle and I open. Now the protein that is supposed to be facing outside is going from the inside the lumen to facing the outside world. Okay? It's you think something like that is pretty complicated in the sense that how it's actually produced, but the Golgi apparatus is able to do all this stuff. The next set of mechanism organelles is the lysosomes. This is your transport and digesting, which is what I call the garbage truck. It basically destroys everything that's coming in and makes it down to recyclable stuff that you can reuse. Lysosomes are very dangerous too because if you activate them inappropriately, it causes cell death. Okay? Lysosomes are also called the suicide sacs of the cell. Okay? And when we talk about apoptosis later on, apoptosis is an activation by certain kinds of compounds that causes the lysosomes to activate and cause release of these enzymes directly into the cytosol, killing the cell. You do not want to cause apoptosis. Okay? That's not a good thing. That's premature cell death. Okay? The lysosomes also catalyze proteins, lipids, nucleic acids, and carbohydrates. So pretty much anything that walks through the door, it cleans up and tries to reuse. There's different forms of lysosomes. I don't need you to go into all that. Just know that there is different forms. There's primary lysosomes that at that point are inactive. Okay? They don't have any active hydrogen pumps. They have normal pH inside the cytosol of that lysosome. When it becomes activated, it becomes what's called a secondary lysosome, or what's called a heterophagosome. At that point, it produces an extremely acidic environment, okay, and begins to destroy things. Okay? And uses an active hydrogen pump, which produces a very low intravesicular pH. Okay? Now, that's really important. Okay? And I can give you another example of that makes more sense. If you look at a human being, okay, the reason that we are able to eat almost anything is that our stomach is extremely acidic. Okay? So we eat, let's say, bad food. If you had meat that's been sitting out for a day or two, and you're a cow and munched on that, the cow would just drop dead. Okay? But you let a human do it, and you'll eat it, and you'll just be happy. You'll actually survive. And you got to say why. And the reason is because our stomach is very different than the cow stomach, which is a ruminating stomach. Okay? Our stomach is basically made to eat anything available, so we're basically scavengers. We're going to eat anything that is readily available. Okay? The low pH kills any bacteria as soon as it enters. So why is that important? Well, if I give somebody an H2 blocker and let their pH rise, what's the likelihood of that person eating raw, bad food and dying? Goes up. Okay? So that's why you see some people that actually go to a restaurant, you see 10 people do fine, and then that one person that's on the H2 blockers dies from bacterial sepsis because the bacteria get past their stomach because the pH was too high. Okay, so the pH in our stomach protects us. On the other hand, we can't eat grass. Okay? We can eat grass all day, but it's not going to do us any good. Okay? But if you gave grass to a cow, cow's very happy. Cow can do it. Why? Okay. The actual pH of a cow is around 7.40. So why would the pH be normal inside of a cow's stomach? What's it doing? It's harboring bacteria, keeping them very healthy. And to add to the bacteria, he basically vomits up his food, which is called cud. He re-chews it, which then puts in more bacteria into it, and re-swallows it, which increases the bacterial count, and that ferments the food, okay? And then the cow absorbs the carbohydrates and the fermentation process from the bacteria. So in other words, if you take a cow and put him into a perfectly sterile environment, the cow will literally die of starvation. He will not be able to process the grass. Okay, kind of cool. Okay, so that's why you understand stuff. Lysozymes are in a sense the same thing as the cows are doing, okay, versus, what humans are doing. We're taking environmental stuff that's coming in, and we're taking it and making it extremely acidic to break down the proteins. So then we can actually use it. We can also use lysozymes to kill bacteria just like our stomach does. Okay? And that's a major concept that I want you to get. Peroxisomes are microbes that contain oxidative mechanisms such as called catalase. The reason we discuss this briefly is when we talk about pathophysiology, when we talk about how oxygen may be toxic at times if you're given too high of a dose. It has to do with production of excessive amounts of free radicals and hydrogen peroxide. So oxygen is not a good thing, okay, if given too high doses for prolonged periods of time. You may ask yourself, well, Dr. Nolan, I remember when I was a kid that the astronauts went up in space and they had 100% oxygen on. And the answer is yes, they did, but at half atmosphere. Okay? And the atmospheric pressure is very important in the sense of number of molecules available of oxygen to cause the injury. If you had somebody on 100% oxygen at 1 atmosphere, you give them about a week and they'll come back with severe pulmonary fibrosis and they will not be a happy camper. Okay? So if you don't really understand that, just imagine that you have your car. Okay? Take your car and leave it in Florida outside for a few years. Okay? What's it going to form real quick? Rust. Okay? Rust is a process of oxidation. Okay? Without oxygen, the rust will not happen. Okay? So you can take that car and go up in high in altitude where you literally will drop the atmospheric pressure and the actual oxygen percentage hits, then the car will literally not rust. So if you have somebody who has got up in Colorado, as long as they're not being affected by salt, their car will last a lot longer than a car here in Florida. Okay? Simply because of the difference in oxygen concentration. If you're a big car fan, you will put your car into an area where it has very low humidity, which is also a bad thing, a good thing, with also altitude, and the cars will keep forever, almost. Okay? So these peroxisomes are important in the sense that they produce these free radicals which actually consume and damage the cell. They will also damage it will kill bacteria as well, so the peroxisomes are also sometimes used to break down substances into harmless products. Okay? The peroxisomes are also important genetically because there's different populations that can break down different levels of alcohol more rapidly. Okay? Our population from Europe, okay, because I'm European descent, okay, our population when we were, our ancient ancestors, we basically took all our water and destroyed it, okay, in Europe, and basically almost all water became non-potable. You couldn't drink it without dying of something from it, okay? So instead, what people decided to do was, we'll take and ferment things and use that as our water source. So nobody drank water. If you looked at Europe in the Middle Ages, nobody drank water. If you did, you were likely to die. You'd end up dying of cholera, all sorts of bad things, okay? So the only thing you drank was beer, mead, which is a honey fermentation process, okay? Or you drank wine, okay? And even to this day, if you look over to France, they'll still see kids being given wine at a very early age, okay? It's all part of their normal, they think water's dangerous, and it is, unless it's, coming out of a good area, okay? So these peroxisomes have the ability to break this stuff down, these wines and ethanol products. So people from Europe have a much lower problem with alcohol intoxication. You're genetically made to tolerate it better, okay? Versus people who never had exposure to it, okay? So like the American Indians. American Indians don't have exposure because they didn't have a fermentation process normally. They would make it occasionally, but it would be something that would not be their daily, everyday content. So as a consequence, people that if you're taking care of American Indians in a reservation or even in a hospital and you expose them to certain drugs, they'll get a much more potentiated effect than you desire. See, that's why peroxisomes are good to understand. Okay. Mitochondria are the next organelle. This is the energy source of your body. It's basically a mitochondria is a bacteria. Okay. What we basically did somewhere in our ancestry is we stole a bacteria, or basically the bacteria became involved with us, or it's a parasite, but it's not a parasite in the sense that it's not causing us damage, but it lives in a very happy cellular environment. Okay, so it's really a host phenomena. We're the host, and the actual mitochondria is essentially a nice friend of ours that produces our energy for us. Okay? The benefit to the mitochondria, which is, if you look at it, what's it look like? It looks like a bacteria. Okay? Almost everything about it is a bacteria. The only thing it doesn't have is what? It doesn't have a nucleus. Okay? If you look at the nucleus of it, okay, it's actually embedded. And one of the things that's rather interesting is we're finding out when we started doing the DNA library, that DNA fragment that designates the mitochondria is very, very similar to one of the ancient bacteria, okay, which is kind of cool. So it most likely is been sort of protected by us by being inside the cell. Now what the mitochondria basically does is it takes oxygen, okay, and it uses that as the end process as an electron acceptor. Okay. The oxygen basically accepts the last portion of the electron transport so it can produce ATP. Okay, so that's why it's called oxidative phosphorylation. So all the major energy source that's produced in the cell is produced by the mitochondria. Okay? The oxidative phosphorylation pathway is the end process of breaking down glucose and eventually using what's called the tricyclic acid or what's called the citric acid cycle to eventually form ATP. The tremendous amount of surface area inside the mitochondria gives it a great area for workload. Okay? And different cells, depending on their oxygen utilization rates, what we call ATP production rates, will tell you how much workload that kind of cell is doing. If you look at the actual myocardial cells, they have a tremendous amount of mitochondria in them. Okay? So that should tell you that it's going to extract a lot of oxygen. And actually, if you look at it percentage-wise, oxygen can be extracted, 70% can be extracted by myocardial cells. That's a huge amount. The normal cell only extracts around 30%. Okay, the brain also extracts a very high amount, almost like 50 to 60%. So those are two organs in your system that are consuming a much of oxygen is delivered to it. The next one is the vaults. Vaults are I found this really, really funny because when I was a student in college, we had a really good class on cellular physiology. And some of us were going into engineering too, which is, I wanted to be at one point, if I didn't go into medical school, I was thinking of becoming a chemical engineer. So one of the things that we always sort of discussed was form followed function. And one of the arguments that we kept on having was it didn't make any sense to us that RNA could actually just come out passively like they were saying out of the nucleus. That made absolutely no sense. How could you get a molecule that's that big and sort of just let it come out, via diffusion? So a lot of us would argue that it didn't make sense. So somebody decided to do some research on it and found out that it doesn't come out by passive diffusion. So if you read that in the past, it's wrong. It actually comes out by a transport mechanism. Okay, and that transport mechanism is called a vault. Okay, it's actually a protein. It's made of what's called a cytoplasmic ribonucleoprotein that's basically shaped like a octagonal barrel. Okay, and what it does is unfolds and accepts the messenger RNA, and then it refolds over it. What's even cooler about this thing is that it binds to another roadway. Okay, which again, it didn't make sense to me how it could passively come out. Instead, it's got a little rail line coming out. Okay, it's part of the cytoskeleton. The cytoskeleton is like an actin filament, okay, that's inside the nucleus that goes out like a road. Okay, and this guy binds to that road, and via calcium, it rotates and comes out into the cytoplasm. So it's got like a road system driving it in and out. So again, this is like a cellular truck. It transports messenger RNA from the nucleus to the ribosomes, or then opens up and then delivers the messenger RNA directly to the ribosome. Pretty nice mechanism. Okay? So, that was 20 years ago. We thought it was just passive. Now we know it's a lot more than that. It's active, and it actually uses specialized trucks. So we still got a long way to go, I think. The cytoskeleton is the next part, which is the road and structural system inside. It's really what's called the bones and the muscles of the cell. It maintains the cell's shape and internal organization. It permits movement of substances within the cell into external projections. It's a network of internal protein filaments, which are microtubules, which can be actin, or microfilaments, which are very, very small. This is what a cytoskeletal structure looks like. This is kind of a very nice, beautiful picture showing you that these things are acting like roadways. Things will bind to them and be transported downstream from them. A typical example of this is in neurons. Okay? Neurons produce their neurotransmitter at the cell body. Okay? Think about this one here. This is a big one to think about. Okay? The alpha motor neuron, which generates electrical activity, let's say to move my foot, okay, originates into my spine. Okay? That's where the cell body's at. The cell body is where the production of the neurotransmitter is at. Okay? That neurotransmitter is, let's say, in this case, acetylcholine. Okay? It has to travel from here, from my back, all the way down in my foot. Just think of that. It's a long way, right? Okay? The way the actual signal goes from here down to my foot is via the electrical signal through the axon. Okay? But the actual neurotransmitter that's being released is released at the nerve terminal, at the end of it. Okay? So it has to be transferred all the way from here, all the way down to my foot. Okay? The only way you can actually do that is if the cytoskeletal structure is intact. So these microtubules actually help the acetylcholine vesicles to actually direct them and transport them down, all the way down to the presynaptic junction, so they can be released and cause the muscle contraction to take place. And so these are pretty important structures. You will find that eventually some disease process affects the microtubules, and guess what happens to the person's ability to contract? It goes away. Okay? So you may hear about some weird muscular dystrophy. It could be something like this. It has to do with a lack of the microtubular system being intact, and then you don't get any more neurotransmitter being released. Now, this is a nice picture of that. This shows you the cell body here. The axon is where the acetylcholine is being produced. It gets incorporated into a vesicle, and then it travels down this chain, okay, all the way down via secretory vesicles, where eventually it gets released at the axon terminal to have its effect. Now, the next, last thing we're going to talk about is centrioles, and I'm going to, at this point, we're going to go on to talk about something else. You don't really have to know so much about centrioles because it's really for division. So I'm not going to go into a lot of detail about this. They're still learning a lot about this area and it may be also involved in aging as well. Now, next topic. So we're now going to the next major heading. cellular metabolism. Okay? Everything that you eat, okay, that you uptake, the body has to try to use, okay, for survival. Okay? How that body does that will depend on your genetics and, of course, on what you're eating. Okay? So when people tell you, you are what you eat, it's to a certain degree true. Okay? The metabolism of different people will be very much genetically dependent. Okay? Some people will consume fats very easily with no problems. Other people will have very low cholesterol intake and extremely have high cholesterol levels. And that's simply because of the way they're metabolizing the stuff. Okay? What metabolism is essentially trying to do is that we're taking our food intake of complex cellular byproducts. When you're talking about plants or animals content, those are cells that have all these proteins, enzymes, everything inside the cytoplasm. And then the digestive system breaks it down, and then we take the essential nutrients that we want, we extract it, put it into our bloodstream, and then distribute it evenly to all the cells, and they use it for food. Okay? So it's a chemical task of maintaining essential cellular functions. If you're anabolic, it means that you have so much energy coming in that you're going to have to convert it into storage. Otherwise, you're going to waste it. Okay? So the way we store things is by converting it into fat. Okay, which is one of the things that most animals do as well. If, on the other hand, you're in a situation where you're in a sense of emergency or something's damaged and you begin to consume too much of what you're taking in, it's considered catabolism. After any form of surgery, the person is catabolic. You cannot reverse a catabolism unless you're like at 6 times the amount of uptake, and most people can't tolerate that. So it's really essentially when you have somebody undergo a major surgical procedure, they all go catabolic. Okay? And basically metabolism is the energy needed for produce cellular function. So no metabolism, cell death ensues. The most important of these chemical energies are adenosine triphosphate. Okay? And adenosine triphosphate is basically 3 phosphorus groups attached to an adenosine molecule that is also of course, phosphodiesterase bond. What you're looking at here is these 2 phosphodiesterase bonds carry more energy, and the third one's the most important because that's one that carries the most energy. So once you break that bond, you basically use that to do whatever you want to do. The nice thing about ATP is you can store it and hold it for long periods of time. Okay, not too long, but not long enough that you can actually store it. So when you don't have enough oxygen, you have that little reserve available. Okay. Once you deplete the reserves of ATP, the cell reverts to forming ATP via either oxygen, or if it doesn't have any oxygen, via oxidative phosphorylation, it goes into an anaerobic metabolism pathway, which produces lactic acid as a byproduct, which is not good, but it does produce a very small amount of ATP. But it's severely limited, and it's not enough usually to maintain cellular life. Now, cellular energy production goes through 3 phases. These are critical for survival, and surgical stress makes patients go catabolic. So you really need to know the basics. I'm not going to go into all the specifics. When you take a biochemistry class, you essentially have to memorize all the enzymes and all that other stuff and how much ATP is being produced. I'm not going to ask you that. I'm just going to ask you the same basic principles, which is how cells produce energy. Okay? The first step is takes food and it breaks it down into essentially 3 compounds. Okay? Proteins, which are amino acids, carbohydrates, okay, or polysaccharides, which are sugars, and fats. So those are 3 things that it's going to break it down into. Okay? Then it's going to convert all these 3 different things into different pathways to eventually form CO2, water, and ATP. That's the end byproduct. Okay? You also have other compounds that are byproducts, let's say, of proteins, which are nitrites and urea. And free fatty acids tend to form more CO2. Now, phase 1 of digestion occurs outside of the cell. The enzymes, these are your proteins to amino acids. When we talk about the GI tract system, we'll go back into digestion a little bit more. Polysaccharides are converted into simple sugars, and fats are converted to fatty acids and glycerol. In phase 2, the intracellular breakdown is the small molecules move into the cell and are further broken down in the cytoplasm. So you have the glycolytic pathway where the glucose is broken down into 2-pyruvate, and then the 2-pyruvate eventually are taken up by the mitochondria, and they form oxidative cellular metabolism where they produce 2 ATP and 2 NADHs, which forms a total of 4 ATP. That's not enough to keep you alive. So what you really need is the tricyclic pathway, which is the next step. The tricyclic pathway is the aerobic metabolism, and you must have a functional mitochondria for this pathway to take place. So anything that causes poisoning of the mitochondria, like cyanide, will basically eliminate this portion of the system. The citric acid cycle, depending on which portion of the country you're on, you're trained in, has also been known as Krebs cycle, okay, or also called the tricyclic acid cycle, tricarboxylic acid cycle, or TCA cycle. And that basically converts acetyl-CoA to eventually form CO2 and water. Now, again, you can get bogged down into all the details. But instead, I want you to think in the big concept, okay, which is metabolism is equivalent to production of CO2 and water, heat, okay, and ATP. That's what metabolism is all about. Okay? Now, what of those can we measure? We can't really measure water content. Okay? We can measure CO2. Very good. We actually measure CO2 all the time in the operating room. We call it the end-tidal CO2. Okay, now you've got that linked in your mind that metabolism is equal to CO2 production. Okay? Let's say I have a patient and I give them becuronium, which is a neuromuscular blocking agent. Okay? So we start the case, we intubate the patient. Okay? We use succinylcholine initially, and then we set the minute ventilation at a fixed rate. Okay? We're not going to change the minute ventilation. In other words, I'm not changing how fast I'm breathing for the patient or how big the tidal volume. It's the same. After the succinylcholine wears off, then we look at the end-tidal CO2, and the measurement says 35. Okay? And now the surgeon says, well, I need the patient to be muscle relaxed. Okay? So it's okay, fine. We're going to give, this is going to be a long surgery, we're going to give becuronium. So we give 10 milligrams of becuronium so the person loses all their twitches. Soon after you see the person lose all their twitches, you see the end-tidal CO2 falls from 35 down to 29. Okay? Most newbies, which will be you, if you don't remember this portion of the lecture, will then take the ventilator and say, you know, Dr. Nolan used to say about the ventilator that if CO2 falls, I need to drop my ventilator rate. Okay? So you'll change your tidal volume, drop it down, and drop your rate down. Okay? Now, my first question is, why did the end-tidal CO2 drop? Okay, what we basically have done is we gave the neuromuscular blocking agent, and when we did that, we basically made that cell go quiescent. So no longer does the muscle contract, and when the muscle is no longer contracting, even though you don't see the person contracting, they are still contracting, maintaining tone. So when you give that muscle relaxant, all that tone goes away. Okay? All that energy that's being consumed to maintain that basically basal tone goes away. So CO2 production goes way down. Okay? So if you change the ventilator, you're doing yourself no good. Okay? All you're doing is you're fixing a number. Instead, just wait. Because now you can use that idea as a concept to how well the muscle relaxation is going to take place. As 45 minutes rolls out, you begin to see the CO2 decline, which means that what's happening to the actual muscle blockade? It's wearing off. So it's a great way of knowing that my muscle relaxant is wearing off. Okay? So don't change anything. Okay? Just leave it alone. Okay? As soon as you see the CO2 come back up, you're not going to try to keep up with it by increasing your rate. Okay? But now you have an essential monitor, which is your end-tidal CO2, is telling you that the muscle relaxant is wearing off without having you doing your train of 4. Okay? This is why we put everything together for you. So you conceptually, clinically begin to apply what you know here. Okay? Now, once CO2 is a byproduct of it, where am I at right now? Okay, yeah. Acetyl-CoA is converted to CO2 and water. It occurs singly in the mitochondria, okay, and this is what we call oxidative phosphorylation. Now, this is the third time I've said that, so you know what? That should be a hint. Hint, hint. Test question, okay? Oxidative phosphorylation. You better know what it means because it's going to be asked, okay? So without oxygen, you don't get oxidative phosphorylation. And in the end byproduct of oxidative phosphorylation is ATP, CO2, water, and heat, okay? It's a final breakdown of glucose, amino acids, and it's basically most fatty acids. This is your tricyclic acid pathway, or citric acid cycle, eventually forming electron transport mechanism inside the mitochondria, which eventually forms water. Okay? There's a great YouTube picture if you really want to see the details on this. I recommend it. Okay? I've given it for the DNAP. I can link it if you guys want to see it. It's kind of a cool thing that actually gives you an illustration of what ATP really does. I'm not going to ask you that in the test, but it gives you a visualization of how complex the mechanism is really for oxidative phosphorylation. Now, the next concept is anaerobic metabolism. Okay? Anaerobic metabolism is taking what's called substrate phosphorylation. Okay? That's the other word for it. Okay? Which it means that it does not require mitochondria to produce ATP this way. This is what happens in the body anytime you don't deliver oxygen to the tissue. So shock states will immediately convert the cell mechanism from an oxidative phosphorylation into what's called a substrate phosphorylation, or what we normally call it in clinical as anaerobic metabolism. This immediately results in the production of very small amounts of ATP, which is only 2 ATP, but at least it's something, and it produces pyruvate, and the pyruvate gets converted into lactic acid. So these people develop a metabolic acidosis. So anytime you see somebody with lactic acidosis, you've got to assume that they're not perfusing their tissue, okay, because they're doing substrate phosphorylation. Let's see where we're at. We'll stop here, and then we'll start back up. A 10-minute break, and we'll start on electrical properties when you get back.